Class, how's it going so far? Because it's kind of like the jack of all trades, master of none kind of a class since it's new. You know, do you feel like it's too fast, too slow, too much information?
Black
Any thoughts?
bacteria's over.
Black bacteria's over?
Yeah.
I won't tell any of the bacteriologists who said that, I promise. But when you tell the immunologist that you're glad that virology is over, then I'm going to have them tell me that, so be careful. But overall, is it like, because it's a survey class, do you feel like you're getting something out of it? And I mean, I guess really the feedback I'm going to be looking for is after my lectures. Since this is the first time, I've taught retroviruses, I know DNA viruses, but I know enough about DNA viruses that it would be impossible to tell you everything in an hour lecture, right? So I really have to pick and choose. So in that regard, your opinion matters. If you think, hey, what I would have liked to hear more about is this, I'll probably have some sort of customer feedback survey at the end to get a feel of what you guys like. Now, what about, are you guys pretty comfortable in the paper discussion format? Because here I'm straddling the fence. I have a little lecture and then a little, or not a little paper, but a paper. What do you guys, who's enjoying like the straight professor stands up here and talks and asks questions versus paper? Who thinks like the lecture format is something that appeals to them a little bit more?
Can we be half and half?
That's fine. Oh, well, you can't raise two hands. I mean, each person has to pick a side. And
I
then
think it's like a happy balance.
Okay.
Too.
Oh.
They're both important.
Okay. Well, then good. I'm balancing quite well today. All right. So today we're going to talk about DNA virus replication. On Monday we're going to talk about retroviral replication. And really, by replication, my mandate is to convey important aspects of the way that viruses replicate their genome, right? Because replication can involve a lot of things, right? There's to achieve a replicative cycle, virus has to enter the target cell, express its genes, do that kind of thing, make more virus. But for the most part, we're going to be focusing really on how genomes get replicated. And one thing that viruses do particularly well is replicate their genomes, right? If you think about it in the context of cell division, now our genomes are pretty big, right? But when we undergo replication, we end up with two progeny cells, right? When a virus undergoes replication, you're talking about 10 to the third, 10 to the fourth, 10 to the fifth copies of a genome in a cell. Scientifically speaking, that's more, right? And that's impressive. And really, we're going to talk about some of the things that make that true today. So what can we notice just about DNA viruses in general? Like what, having maybe thought about viruses for a while, what's the first thing I think about when I think of DNA viruses? Well, so first, since I'm a cell biologist, and usually the viruses with the most genes will teach you the most about cell biology, I think of the fact that there's two of the really big, heavy-hitting genomes in terms of genome size are DNA viruses. You have your herpes viruses, your vaccinia viruses. These viruses, in terms of genome size, dwarf other viruses, and Epstein-Barr virus as well. So all the herpes viruses are universally huge, right? Pox viruses are huge. Compare that to like a retrovirus, 10 kb, right? And in there, a lot of it we'll talk about is kind of LTRs, where it facilitates replication and there's no genes. So the genomic capacity of DNA viruses is monstrous, right? And another thing that we're going to see, and we won't talk too much about viral polymerases, but in many cases, if not most cases, the viruses take advantage of the fact that we already have the ability to do this, right? We already have DNA replicative machinery in our cells. And so in a lot of cases, the viruses want to, evolution will always drive a virus to be very efficient, and that efficiency will make, one protein have multiple functions, but will also drive the reality that if you can get it from the host cell, why are you making it, right? This is so much easier than you're making it, is kind of what Darwin would say to these viruses. So a lot of the DNA viruses use the cellular DNA polymerase machinery to replicate their own genomes, right? And even those that are viral, right, if you were to align the structure, and I'm sure that you guys got worn out and maybe in polymerase structure in a core course last semester, but the basic core module of the DNA polymerases that viruses possess are very functionally analogous, and you can align them with our polymerases in most cases and see that somewhere way back in the primordial soup, viruses didn't invent that. Or maybe viruses invented that and we stole it, right? But minimally, there's a common ancestor or theme among the polymerases that allow the viruses to achieve this goal when they have decided to make their own, right? If you compare that to retroviruses, which we'll talk about on Monday, reverse transcriptase is a very retrovirus specific thing, right? We never really had that idea. The central dogma of DNA to RNA to protein, is something we were very comfortable with for a while now. But retroviruses are like, what if we put a weird twist on this? DNA viruses, there's no weird twist. These are things that have been done before, right? And so, with regard to that, and this is kind of the theme of the last slide. Can you guys see this up here? It's kind of up high, sorry. But so DNA replication, there's an app for that, right? So again, the things that you've learned about DNA replication in, what class would that have been? I'm sure you've learned it, right?
Mm-hmm.
Can one really avoid DNA replication in the core courses, or have you not had it yet? Okay, right, you guys got worn out, you guys had enough, you don't want any more to eat in that regard, right? But there are certain realities that are still going to be true for these DNA viruses as they replicate their genome, right? Just like eukaryotic DNA polymerases, all DNA synthesis is template directed and occurs in the 5' to 3' direction. Viruses are very innovative, but there's not been one discovered yet that goes 3' to 5', right? So that's like a universal law of physics, even though it's not physics, right? Number 2 is parental strands are copied in a semi-conservative replicative mechanism. What does semi-conservative mean? I forget that word all the time.
After the parent cell goes, or parent strand goes to the daughter, and the other half stays with the parent.
Exactly. So it's one parent strand, one new strand in each of the 2 progeny copies of the genome. The replication of DNA begins at specific locations. And actually, so this is one of those things. So what's definitely true about viruses in general, and especially DNA viruses with these huge genomes that will have, we're not going to talk about the accessory proteins of DNA viruses today, but if you're a cell biologist, that's why you really love these viruses with huge genomes, because they're tweaking a pathway that you might not even be aware of, right? You start studying this protein, and all of a sudden you discover a pathway, because you're like, hey, this virus thinks this pathway is pretty important. Why is it doing that, right? Well, the discovery of origins of replication really were first performed in viruses. We'll talk about SV40 in a couple slides. And really, that's the first, to my knowledge, the origin of replication that was understood in a non-bacterial system. People are still trying to figure out, so our chromosomes have ories, right? But really pinning them down and figuring out exactly how they work has proven much more difficult. Right, so this whole idea of ORI recognition really became first established in DNA viruses, even though DNA viruses are doing much the same thing as we are when we replicate our genome. And then, this is kind of like a duh thing, but they had it in the book, so I put it in here. But so the synthesis carried out by DNA-dependent DNA polymerases, right? So if they were DNA-dependent RNA polymerases, they'd be transcribing stuff. We wouldn't be talking about virus replication. And if it was RNA-dependent DNA polymerases, well then it would be a retrovirus. But all right. So what I am going to focus on a little bit today is template priming, right? So if we just kind of skip the DNA polymerase aspect of this and accept the fact that once you get going with a strand of template copying, that you're going to obey the laws of base pairing and things like that. The ways that viruses, the DNA viruses, are somewhat diverse is the way they prime their DNA to get that initial polymerase reaction going, right? Because polymerases don't just say, oh, look, there's single-stranded DNA, I'm going to copy it. And often they need help separating the 2 annealed strands of DNA to gain access to the template, right? And so the ways in which DNA viruses prime that reaction varies considerably, and that's worth some consideration. So what we're going to see is that there's viruses that are primed using RNA priming. So there's a little RNA sequence that will prime and become that will base pair with the DNA template and that little bit of RNA primer will be extended out as a DNA strand by the polymerase, and we'll talk about SV40. So SV40 also, just in terms of history, like sometimes in the current literature, what does get lost is kind of the things that were historic or epic, and really the understanding of SV40 DNA replication really preceded the understanding of eukaryotic DNA replication in many ways. There is DNA priming. So, and what that means is some viruses have little kinks in their genome that will kind of wrap around and prime themselves. And we'll talk about a pretty demonstrative example of adeno-associated virus doing that.
So can you say again for DNA priming? So the virus use its DNA to wrap around the cellular genome, right?
Right. So, I mean, essentially, and we'll see a slide in a second, but this really all comes down to palindromic sequences. So AAV has a stretch of DNA. Maybe I can even color code it here. Fancy. So there's basically one palindrome that forms base pairing here, and then another palindrome that extends off this strand that forms base pairing here. So you have a free 3' end. So this is one contiguous stretch of DNA, right? Mm-hmm. And then, but now you have this exposed 3' end of the DNA. And this is the perfect primer to allow a polymerase to extend that. All right, and then finally there's a protein primer. So the viruses that prime using a protein, there's only a couple of DNA viruses that do this. Perhaps in your in the other lectures you'll hear about other proteins that, or other viruses that get primed with a protein that aren't DNA viruses. But the case that we'll talk about today is going to be adenoviruses and protein priming. Okay, so the first idea that I think is worth discussing I want the opposite of that. How do I turn the lights up here off? Is it over there?
I think that's the only switch.
There's really no switch for this? Fine. If you guys fall asleep when I turn the light off, you'll hurt my feelings. I guess that's fine. So, in the case of a lot of DNA viruses and the SV40 virus that we'll talk about, what you have is an origin of replication, and this origin of replication is critical for initiating something called a replication fork. And a replication fork is very interesting because it looks like a spoon, but they call it a fork, and I have no idea why, right? But what you can see here is that you have the separation of these two original strands of DNA, and then the semi-conservative replication of these two strands, just like you saw in DNA replication when you took the eukaryotic cell biology class, right? So just like eukaryotic genome replication, DNA viruses use their replicons to replicate their genome, right? And so what's a replicon? A replicon is, there's these distinct units that are in a viral genome or a eukaryotic genome, and these origins initiate DNA replication at different sites, right? And that's really key. So I think it's in the book, but they talk about in eukaryotic genomes, if there wasn't multiple replicons, if we just decided, hey, we're going to start replicating our DNA, you know, they tell you how long it would take to finish replicating your DNA, but it's something on the order of like years or something, right? So by multiple origins, by starting the project in many places and then connecting, you can achieve cell division faster than that, which is huge for us. And viruses are no dummies. They do the same thing, right? And so each replicon has a single origin of replication, and replication proceeds away from the origin in a bubble-shaped replication fork. And, you know, do I have much more detail on this? So I won't go into much more detail on this, but well, essentially here's an electron micrograph of one of these bubbles kind of enlarging. So you can see how DNA progression would begin with this little itsy-bitsy bubble here, kind of progressing forward to achieve two double strands of DNA. And in the same way that, you know, there's a leading strand in a replication fork that is 5' to 3' and continuously synthesized, and on the lagging strand, there's discontinuous replication that has to be, these segments have to be combined. All of this is true in viral genomes, just as it's true in eukaryotic genomes. So I'm going to assume that you've been efficiently taught and worn out on this topic. So we're going to move forward. So I bring up the the simian virus 40 ORI. Number one, because it's a classical ORI in a system that people have studied for some time, but also because we'll see either in the lecture or in the exam that there's a real world application of why we should care about the SV40 ORI that most people often forget, right? But like anything else, there's recognition elements that facilitate the protein binding within a DNA sequence, right? And the one thing that's for sure, if you haven't noticed this, whether it's bacterial proteins, viral proteins, or host cell proteins, they love palindromes, right? Palindromes help recruit proteins to DNA molecules, and the SV40 ORI is no different. And so SV40 is one of many DNA viruses which rely on cellular enzymes to replicate their DNA genome. But that's not how it starts. It starts with the recognition of the ORI by the SV40 large T antigen. And this is one of the first proteins that the virus produces. And if you look at it, it actually forms this barrel-shaped hexamer where the DNA, the double-stranded DNA helix, runs down the middle of, right? And this is critical because what SV40 helps do is to unwind the DNA with the help of another viral protein or a cellular protein, RPA. And by unwinding the DNA, it begins this process of forming a replication form, okay? So there's a few examples here where, and in the book there's a lot more detail provided. I just want to make it clear that, what I'm looking for you to remember are the big ticket things. If you're remembering the last little bit, I will not ask you a question about FEN1, okay? Because this is really true for SV40 and probably not other DNA viruses. Our job here in an hour is to cover the big ticket items of DNA replication, right? And those big ticket items are here as a classic example of DNA polymerase alpha, which is one of our DNA polymerases, acts as a primase in this system. So it is going in, it is priming, generating an RNA primer, and it acts as the initial DNA polymerase. But following the formation of this RNA primer, at that point, other DNA polymerases are recruited and begin the full-scale replication of the genome, okay? And so, and again, this is one of those things too that, this should be pretty obvious to you guys by now, but when you have these free 3' hydroxyl groups on DNA, right, there's a lot of proteins that want to kind of jump on that bandwagon. And by generating these 3' hydroxyl groups, this recruits cellular proteins that synthesize the leading strand of DNA, and the lagging strand, again, is replicated as we talked about. But you have your proliferating cell nuclear antigens, some replication factors, and a DNA polymerase. Okay, so again, this is a classic example of a virus knowing what it wants to do, right? And there's always a couple subtle differences between the viral genome and the eukaryotic genome, right? But really, the heavy hitters in this regard are going to be the large T antigen and the viral RPA protein. After that, it's very convenient for the virus to use the host cellular machinery. Now, and I almost put a slide in on this idea, but it was just, the amount of material I think we're going to try to cover here is pretty extreme today. But so since viruses, DNA viruses especially, use the host cell DNA replicative machinery, there's another pretty common feature of DNA viruses, especially those that use cellular polymerases. They have a very specific effect on cells. Can anyone guess what that is?
if they're using cellular polymerases, they're taking those polymerases away from replicating the cells on the stuff.
Sure, but let me ask you this: what do you think now in most of your cells that are just sitting there? How much DNA polymerase do you think you have in those?
Probably a lot.
Well, but no, actually, I would argue that the opposite is true, right? In your cells that aren't dividing, you don't have a lot of DNA polymerases, and maybe some of them might be high. So there's probably certainly examples where you're right, but certainly the whole congregation of enzymes that one needs to replicate DNA is enhanced, rightfully so, during cell division, right? So there's a lot of DNA viruses that have a vested interest in facilitating cell division, right? And this is kind of an off-target effect, and I wasn't sure if there's like an oncogenesis or a virus control of cell division lecture later in the semester. But there's a number of examples where viruses, especially DNA viruses that want to use this machinery, they kind of go in and they tickle the regulatory mechanisms of the cell that control cell division. And they just kind of turn it on, right? So that gets all the genes that they enjoy upregulated, and then they can enjoy the benefits of those. Now they're like, well, that's probably not good for the cell, and right, no kidding, right? And by causing uncontrolled cell division in some cases, that can actually be the more negative aspect of a viral infection. Okay, so, and this kind of like, I would say in general, right, this kind of DNA synthesis using the replication fork and mechanisms like this are common, especially among the huge viruses. Adenovirus actually is pretty big. We'll talk about a different mechanism there, but usually those big viruses, they enjoy a replication fork and multiple ories because I think in terms of generating these large genomes, right, it's the same mechanism that we use, and we have pretty large genomes ourselves, and there's a reason for that. Okay. But now, let's talk about something completely different, right? And this is going to be structure-based DNA priming by AAV. I think that was a pretty good drawing. All right, so now the key to this is one of those interesting stem loop kind of things that is formed when you have single-stranded DNA. And so I would say the classical defining feature of the AAV genome is these central palindromes that are flanked by longer palindromes that form these little T-shaped ends, right? And what you can see here is that in this case you have a palindrome here that facilitates this T portion, and then this other palindromic segment that kind of primes replication in this direction. Now, you'd think to be more efficient, you could prime replication in this direction. How come that doesn't occur? Thank you. Amen. Right? So, no, sometimes I ask trick questions to see if you're paying attention. And they're right. That would break the rule that we talked about earlier, right? Which is 3' to 5' is not legal, right? Which you'd think somebody would have invented it, but I guess whatever. But it never goes from 3' to 5', and that's the kind of extension that would be, right? And so, if you start out with these terminal repeats, they form these structures, right? And then what you have is the extension of this DNA. And so something really interesting happens when this when you get the initial replication of the viral DNA here. And when I explain this to you, think about how valuable this would have been if you were actually the person that's kind of first characterizing AAV replication. Because, you're getting your sequences back, and you're like, what the hell is going on? And so what am I talking about, right? So during replication, how do you get, if you just start priming here, how do you get to this other portion of the genome that you'll obviously have to replicate, right? Well, there's a specific viral protein that, again, do not be remembering rep 78, rep 68 for the exam. I'm really not like that, okay? But what it does is it nicks the DNA at what's called the terminal resolution site. And this nicked fragment now acts as a primer, okay? And this primer can also facilitate the extension of DNA that will now utilize this fragment here as an unwound template. Does everyone see how this would kind of flop open and then you could polymerize that to get this, right? But look at this. Is this the same as that? But it's pretty darn close, right? But now if you were trying to figure out what's going on there, right, you're sending these things out for sequencing, and you don't know what it is, but you have some primitive DNA strider alignment, or maybe you're even more advanced than that, and it's coming back, and sometimes it aligns perfectly, but sometimes there's these interior fragments that don't make any sense, because sometimes it's ABCAD, sometimes it's ACBAD, and you're like, what? Right? But so, I think getting this data back, would you think that's you? Would you think that's your fault? Be like, oh, I'm screwing it up. It keeps coming back 50-50. I don't know why that's happening, right? Or would you be like, this is really telling us something about how the virus is doing this, right? Because that's what the answer is, right? Because in a lot of cases, the orientation of these genomic boundaries is absolutely required for maintaining the replication competence of a virus. Now, what we'll learn about on Monday is you can't go moving around these terminal elements of a retrovirus and still have a retrovirus, right? That would screw everything up. Adeno-associated virus, apparently it doesn't matter, right? Because the gene or the population of adenovirus, while it's replicating, will then be a combination of these two different ends, which can be ABCAD or ACBAD. Both are replication competent. And if you think about it, when you kind of go back to the beginning, if you start with ACBAD, that replicative cycle will bring you back to ABCAD. Does everyone get that? So in a replicating AAV culture, there's no right answer of what does the genome look like, right? It's 50-50. But the virus has kind of evolved a way to make this not necessarily deleterious. Normally you can't just be shuffling the parts of your genome and have that be an effective replicative mechanism, right? That's kind of unusual. And the people figuring this out, they really had to step away from the box that they were trapped in to probably figure this out, right? Because I would be sure that was us screwing something up, at least for a while. But once you have this hairpin formed off of this primer, now you can form this structure again in something called strand displacement, which I think from the cartoon it's pretty obvious what strand displacement occurs, and that facilitates the replication of another full-length genomic segment. Okay? Going here. Okay, so we're going to go through protein-based priming by adenovirus. And so what does that mean? So, really quickly, so there's a serine on a viral protein that gets conjugated to what is essentially a small little oligopeptide, if you want to, or no, oligonucleotide primer, that then, by conjugating it to the protein, it can act as a primase, if you will, to facilitate the polymerization of the viral genome. And in this case, so the polymerase acts to actually replicate the DNA, as you might expect. And so what do you think this is here for? What is that? This yellow stuff that's coating this piece of DNA. And why would you need it?
For marking. For marking.
Louder.
For marking. To make a mask, like to know distinguish between two strands, right?
Well, sure, but this strand here is kind of sitting around flopping all by itself, right?
DNA binding proteins?
Exactly. And so why would you have a DNA binding protein on this little strand here?
So it doesn't come back together?
Right, either prevent it from coming back together or also single-stranded DNA. One of the big problems, right? So again, we didn't really have time to go into this about DNA viruses, but there's certain things that just will freak cells out, right? Number one is DNA in the cytoplasm, right? So DNA viruses either have to pretty stealthily get to the nucleus, Or in the case of vaccinia viruses, they form these pretty closed compartments where it's just them and their business in the cytoplasm. But DNA in the cytoplasm will freak cells out, right? Another thing that freaks cells out is single-stranded DNA, right? Single-stranded DNA shouldn't be there, right? And so by kind of covering this DNA, this single-stranded DNA with DNA binding protein, I don't know that there's been a paper showing this, but maybe before you all graduate or 10 years from now, there'll be a paper that comes out and be like, oh, Campbell, you were right. There's some there's some cellular mechanism that is built in, ready to recognize single-stranded DNA and thwart viral replication. So you kind of cover it up. It's an act of stealth, right? And I think really the protein-based priming idea and the DNA binding protein are the things that I would take from this slide. From there it's a pretty basic DNA replicative cycle. Question?
Can you repeat how the serine conjugates and that whole part of it?
Yeah. So to this cysteine, there's just essentially a DNA oligonucleotide that gets conjugated onto this cysteine via mechanisms that frankly I'm not sure of. But then since you have this little oligo here, now this can anneal to the genome, right? And then normally you synthesize a primer, or another common mechanism is to take an existing primer, like so HIV uses a tRNA that just kind of anneals to the viral genome, but you need something to get it started, right? And in this case, it's going to be this little short segment. Okay. So are there, that's about what we have time for. Are there questions? We can certainly have a little bit of a discussion. If we don't finish the paper, my feelings won't be hurt, and I think I can jam through it if we're running short on time.
Is that a viral polymerase or? Polymerase?
Yeah, I believe so. Yes, in fact, I'm pretty sure. Yeah. Okay, so if you read this paper, the reason I chose this paper is because there's a lot about it that I like. Right? Well, number one, there's a lot of picture taking, and I'm a picture-based guy, and I think you can see a lot just by looking, right? Number two, what it really emphasizes is a fact that is true about really almost all DNA viruses, especially those guys with slightly larger genomes, is that diffusion is bad for a process like this, right? So when you want to replicate your genome, and especially, I'm not just making one copy, I'm making 30,000 copies, right? To do that, you require geographic efficiency, right? And what do I mean by that? It means that locally, you want your concentration of proteins to all be in a row. You can't be waiting for a protein to diffuse out of the vast nothing of the cytoplasm or even the nucleus. You want to have all of the proteins involved in your replication in a somewhat constrained area, right? Or a replicative compartment, if we want to use a buzzword for that. And replicative compartment is something that DNA viruses do both in the nucleus, in the case of what we're going to talk about here. Again, vaccinia virus does it in the cytoplasm. But making these replicative compartments is what allows viruses to be so explosive, right? Because that's how you make that many copies of your genome in a short amount of time. Anything else would probably be bad for business. And so one of the things we'll see today is that that's exactly what herpes virus does. And so to a certain degree, a lot of the data in this paper is looking at a phenomenon that was already established, but we're going to kind of see it for the first time. And that's going to be the idea of these replicative compartments in the nucleus, right? And number two, there's this idea of this host cellular proteins that are, number one, involved in facilitating viral replication. And also, number two, the way that you interpret the data. My punchline for this paper might be very different than what you think. And I guess the last reason I like it is, I mean, what did everyone think of the first paragraph or so? First, the lead up to the first picture. Did anyone notice anything there in the way they were describing that data? Because, I mean, part of the fun of being in science, right, and being in scientific battles, if you will, is, who doesn't love a good scientific beatdown, right? And you don't have to know always the backstory, but read that first, paragraph or two, because that's about as much of a trench war as you can get in, right? Because clearly somebody else said something about one of their antibodies, and they just went to town on them, right? They're like, they said it was this, and we're going to show you in really definitive fashion just how wrong they were, right? And those are the kind of things, you never want, if you're going to be wrong about something, you want to be wrong about something that no one cares about, right? Because otherwise, people are going to have something to say to you. And that's clearly what happened here. But so, does anyone want to explain this first figure? And maybe I can give you this, or I can just grind through. What do we feel like we want to do? Because I'm not looking to drag you guys by the nose through this. But at the same time, if you read it and you want to talk about it, I don't want to cheat you. You guys talk too much. Okay, I'll roll. All right, so what they're first going to do here, and the other reason that I think I like this paper is that it shows you how funny immunofluorescence can be, right? And how you make certain assumptions, and when you make certain assumptions with some sort of immunofluorescent experiment, oh man, that you're going to look back on that day when you assumed, and everyone knows the scientific euphemism for assume, right? What happens when you assume? Everyone knows that. Make an ass out of you and me. All right. So what they're doing here is they're characterizing a couple of antibodies, right? I think these are mouse monoclonals and maybe these are rat polyclonals, to look at the localization of PML in cells. So what is PML and why do people care? Right? Because this is a pretty interesting thing too, and when we get to the punchline, I guess we'll be interested by the punchline. Who can tell me what PML is, either historically or why people care? Well, I will tell you.
PML is just like polymonoleukocyte, right?
So it's a promyelocytic leukemia protein, and that's how it was first characterized. Go ahead.
It's a protein contained within the nuclear domain.
Sure. So there's nuclear bodies in a nucleus that people still don't understand very well, right? And even the understanding that is generated in this paper, I'm willing to bet $5 in another direction, although they kind of touch at it in the discussion. But there's these nuclear domains in the nucleus, obviously, right? They're called ND10 domains, and they're accumulations of protein. They're not quite nucleoli, but they're protein-dense regions that have things like PML, ND10, and SUMO. And importantly, where PML first comes from is there's a very classical genomic translocation that occurs in people with a very specific form of leukemia. Okay, so if you fuse a portion of the retinoic acid receptor to PML, what you end up with is a oncogenic protein that causes a leukemia, which is why this protein is called PML. Which I guess maybe I'll risk running the punchline, because if it wasn't called PML, what's its other name? Does anyone know the secret identity of PML? It's kind of like a superhero kind of thing. No? Oh, you almost said it. What was it? Oh, you said something that started with the T. So PML's... Exactly. PML's TRIM19, right? And they kind of, in this... When was this paper published? Like 2001? In 2001, they kind of allude in the discussion that maybe this TRIM protein is acting in some antiviral fashion, even though we don't have it figured out yet, right? Well, in the last 12 years, what people have come to understand about the TRIM family of proteins makes that a pretty easy bet, right? But here, they're looking at it as, the PML is getting recruited to these replicative compartments because it must be needed. The virus really needs it there, right? And more appropriately, probably what's really happening is PML is trying to exert some sort of antiviral effect that's being thwarted, right? But that doesn't, how could they know that at this time? And what's certainly true is that there's these disruptions of the ND10 bodies that's required for virus replication, which we'll talk about in a second. But it's fun to look at this data because, this paper is very well written, and the interpretations in terms of what can we say, it's very rigorous, right? These guys are wonks and masters of their crafts. They're not like doing one antibody and guessing, right? If anything is true about this paper, is they're being so careful and methodical, it kind of makes our head hurt, right? But even they don't, like, they're being very careful, but they would love to know the thinking of 12 years in the future, because I bet you that the way they're thinking about this problem would have been fundamentally different. But okay, with that being said, we got some figures to get through. Okay, so first what they see is some basic co-localization, although it's 2001 and they don't have that great of a microscope, we'll give them a pass, right? But what you can see here is that ICP8, what's ICP8?
Binding protein binding.
But it's a viral protein,
Yeah.
right? And so what they're going to use are these viral proteins as markers of the replicative compartment of HSV. So what you'll see invariably is that most of these proteins will go to the nucleus, but more importantly, and what you want to focus your attention on, are the fact that there's areas of increased density or accumulation within the nucleus in infected cells, and those are these replicative compartments, okay? And in some areas, they actually kind of blow them out and they ruin it because you can't really see the replicative compartments here, right? This is the danger of pixel saturation that we talked about in the fall, but, what are you going to do? Here, it's a little bit lower exposure, but you can see nicely here in this cell, you can see the co-localization of PML to these replicative compartments of the herpes virus, right? So we have 4 different antibodies, right? 2 of them show this localization. And then, okay, well or 3 of them show this localization, so then what's up with that, right? And that's the first thing you should ask. But also as an aside, I put this up here as kind of if you're doing a journal club or you're presenting your data or something, can everyone see this pretty well? Can anyone see it better now? Right. So if you're going to put pictures up in the journal club, I don't know if they teach you black backgrounds in the presentation course, but boy does it help, right? But this is just the same figure. And so we don't want to focus too much on this. This is kind of settling the fight and really characterizing what antibodies are recognized by what. But the bottom line is that PML has a lot of splice isoforms. And so there's these exons 1 and 2, which are the invariable exons. And these invariable exons probably encode that tripartite motif. If you guys are trim wonks, there's a ring, B box, and coil there, right? But after that, there's all kinds of variability in the downstream splicing possibilities of PML. And what they have is 3 cDNAs of 3 different splice isoforms, and what they're going to do is figure out which antibodies are most appropriate to use. And this is always, again, the reason I think this is a good paper for you guys is because at some point you'll be given an antibody, and the assumption will be you're going to go and look at your protein, right? And maybe that'll work, and maybe it won't, right? And when thinking about what went wrong, rarely do we think at this level of depth, right? Normally we just blame Santa Cruz and move on, right? But sometimes that really doesn't help us get any further. And it's useful to think about the science that might underlie our failed result or our successes, or why somebody else disagreeing with us, right? Oh, they used this antibody to PML, I used this one, but I'm not going to really pay attention to that. I'm going to pay attention that they disagreed with my paper, and now I hate them, right? Because they're my enemies. Well, maybe they just used a different antibody that recognized like a different splice isoform. Is that possible? Don't talk to me. I'm angry at those people, right? Because scientists like them. If you don't know them yet, you'll meet them, right? But sometimes there's actually science in these little feuds that, if you guys take the more advanced virology course where we talk about restriction factors, I have another really great story in that regard, and then the answer ended up being Apopec 3G. So what they're going to do is they're just going to map these antibodies, right? And by immunofluorescence, they're going to see that this 5E10 antibody, which other people probably had used and said things that were mean, be like, oh, see, this proves that so-and-so is wrong. Well, it only recognizes this, I believe, 7th exon of the protein, right? So all of the other PMLs are not recognized by this 5E10 antibody, but this PGMG3, which they'll use for the rest of the paper, rightfully so, it was raised against exons 1 and 2, or that part of the protein that is not variable. So lo and behold, it recognizes those domains in any splice isoform that exists, right? So that's nice. So they'll be using this for the rest of the paper. And this is kind of like the last, and so like this paper is kind of this transition, right? Because sometimes you have to squeeze kind of two different stories into a paper. This is the last, public squashing of the people that probably said that they were wrong about something. And then they're going to transition to more mechanistic studies. But what are they doing here? So first, and this is one of those things where, negative results in lab, like things that don't work, right? Our tendency is to just throw those things away. To say like, God, I never want to think about that again. That didn't work at all, right? And actually, so the protein that I work on, TRIM5, kind of came from that, right? Because people were trying to make monkey models for HIV pathogenesis studies. They wanted to find monkeys that they could get sick. And the people that found monkeys that could get sick, they took those monkeys and ran. There were other monkeys that couldn't get sick at all. And they're like, those monkeys didn't work, right? And then years later, people fished out TRIM5 alpha from those monkeys that didn't work. And people are like, oh yeah, that explains that, doesn't it, right? But so here's another bit of lemonade that can be made in that regard. Maybe not quite as big. But at some point, somebody was trying to stain Vero cells and look at PML. And they noticed, man, I don't see anything, right? And that's probably because the form of PML that Vero cells are making, I think it's a monkey cell line, aren't recognized by this antibody, right? And you're like, oh, I guess we can't do our studies in there, right? Or the other way to think about it is you kind of have a knockdown cell line for PML. So you can throw in there other PML isoforms from the species that your antibody does recognize, and now you can start to see where that isoform and only that isoform go, right? Now that wasn't a huge logical leap, but I can, from the people that I've met, not too many people would be able to keep this idea in their background RAM spinning long enough to turn it into something useful, right? We tend to want to forget these things. Well, so what you can see here is they kind of categorize the localization of these different PML isoforms. Now what's true about this? You don't even have to, is this going to localize to ND10 bodies or replicative compartments? No, it's not even in the nucleus, right? You can actually kind of see where the nucleus is in these cells by looking where the protein isn't, right? Now, maybe in a cell, this might be a really rare isoform that would have been hard to look at exclusively, just look at this isoform and say that it doesn't go to the nucleus, but now that you have this background cleared up, now you can ask these kinds of questions. And what they see is that really the only one that does go to these replicative compartments, although this is probably the weakest imaging in the whole paper, is this PML 3467. But that's really not why we're reading this paper, so I'm going to...
I have a question.
Go ahead.
That's the same pictures, like, they say that PML3467 is like working, but not the other PML1367. But I saw the picture. It looked like the same. But how come they can make a conclusion like that?
Yeah. So of all the conclusions that are weak in this paper, I'd say that this is the one. So what they want to say is that this PML isoform is localizing to these replicative compartments. So there's, I'd say, two decent examples. So if you can kind of look, there's a slight bit of increased density here and there that is probably, it's definitely apparent there. It's probably apparent there, but they've saturated their pixels. So I think it's true, but I think the evidence, as you say, is pretty rough. I also think, so there's probably an increased density here, because you can kind of see it here. But now, and this is that, in the methods course, we talked about how if you oversaturate your signal, you will lose the resolution, right? Well, this is the classic example of that. Because when you can't see differences in pixel intensity, this whole thing looks red. In other images that they show in this picture, you can see the differences in staining intensity that help you appreciate the replicative compartments, right? So this is a perfect example of the reason, that you have to be aware of when you're saturating your image, because this is the danger of that. But so here is really, I think, the definitive thing, and even getting to this picture is the moment that we needed to do, because this brings home the idea of these replicative compartments. Does everyone see that? Right? So here's the nucleus as a whole, and here is much better imaging, right? And it's much closer too. If they would have, if they would have brought these nuclei into this level of focus or this level of magnification, I think you'd probably be more compelled. But it's one of those things maybe they didn't want to go back and do. But here, what you can see is these viral proteins don't just accumulate in the nucleus. There's these pronounced areas of accumulation, right? And PML is also there. Now, also keep in mind, this was really 1991, so these are 2001, but these are two different cells to try to demonstrate the same thing, presumably because they had a two-color fluorescent microscope, right? It would have been much easier in our world, right? So why don't you just do 3 colors at once? But first they looked at the correlation between the viral protein and PML, and then they look at the ability of the viral polymerase to be recruited to these structures, right? And the role of the role of the viral polymerase in expanding these structures is also very important. And we'll see that in a second. So here you have exonuclease defective viral polymerases, but that really doesn't stop the replication, right? And you get these large replicative compartments that contain both PML and the polymerase and the viral protein. That's pretty compelling, right? Now, and this is pretty interesting. Now, if there's no polymerase produced, right, ICP8 will kind of nucleate some little, probably, organization of the existing viral DNA and maybe some host cellular factors, but you don't get this expansion into replication centers, right? And then, the PML localization is lost, and obviously this is background staining for polymerase, right? Now, if you have an active polymerase, but it's still there, can it get recruited to these sites of ICP8? Not really. So there's something about the activation of the polymerase that maybe the activation of the polymerase draws ICP8 into different foci, right? But again, the localization of PML to ICP8 is not occurring. So something about the virus actually replicating is telling this cellular protein where to go. Now, if you read the paper carefully, that's not their hypothesis, is it? Right? Their hypothesis is that, PML is not getting recruited there, and maybe that's the problem, right? But, with 12 years kind of to reflect on the realities, my position would be that there's no problem, so PML isn't recruited there, right? But that's one of the ways that we have to think about these data historically, is that people didn't know as much way back when, like 2001. And then, so let's see, here's a temperature-sensitive mutant at the non-permissive temperature. And what you can see is you get some ICP8 PML co-localization. Kind of ugly pictures here, but sometimes what you look for is the pattern. Have you guys seen like The Beautiful Mind, at the Russell Crowe movie? You can kind of see this little U-shaped thing here that's also there. It's not as beautiful as you'd like it, but it's there, right? And then also there's areas of co-localization between ICP8 and polymerase. So polymerase is now getting recruited, and something about perhaps its ability to become active as opposed to this inactive polymerase. This is also inactive at the non-permissive temperature, but now it's getting recruited to these replicative compartments that are just starting to form. And these details of which mutant goes to the ICP8 foci, again, this is something that I'm not going to test you on. But what it's useful to do is maybe in a survey class like this is imagine how these complexes are going to start coming together, right? You have a little piece of genome. You have an organizational DNA binding protein. And it's going to start creating some structure that will facilitate the scaffolding on of these other molecules that ultimately give you something like this, right? And what's going to happen in that context? It's going to be viral proteins kind of starting something, viral genomes starting something, but then it's going to require the recruitment of cellular proteins that are going to facilitate that. It's going to probably induce the recruitment of other surveillance proteins that are going to be like, hey, what you doing over there? Are you a virus? Are you replicating DNA in my nucleus? What's going on? Right? That's probably what PML's doing, because in the later figures, and maybe we won't get to it, but we can do antiviral strategies at another time. That's probably what PML's doing. It's not getting the job done here, right? Something about disrupting the bodies, like, well, herpes has another protein, ICP0, which is specifically designed to disrupt PML bodies. And if you don't disrupt those PML ND10 bodies, you don't even get this far, right? But they're just thinking, oh, they've got to get PML out of those bodies so that they can come help us, right? Well, I don't know about that. So here, again, we're going to look at the localization of polymerase and this ICP8 in cells. And the punchline here, we're going to, I guess, move pretty quick, is that, in a good replicative compartment, you have good, strong co-localization of the viral protein and polymerase, right? But if there's no polymerase produced, the whole thing doesn't get started, right? Obviously, if there's no Pol produced and you see Pol standing, that's a big problem, right? But the important thing here is now you don't see those similar structures in ICP8. So it's the recruitment of these larger protein complexes that are going to facilitate replication. And when you do, you're looking at a structure that doesn't include a few molecules. You're looking at essentially a factory. I think we can skip the rest of that. And this was this was one of the fun things. And this actually, I'm not going to lie, one of the reasons I like this paper is because it helped us when we were working on TRIM5 a little bit to have the courage of our convictions. Because so here are these pre-existing ND10 bodies or PML bodies, right? And what these authors observed was, number one, when they added a proteasome inhibitor, MG132, you didn't necessarily get more of them, but they got brighter, right? And this is brighter, at least in 2001. This is, I guess, you could do a little bit better nowadays. But they're brighter puncta. And also, arsenic seemed to do about the same thing, give you brighter puncta. Now, why did they test arsenic? Because this was really cool. Scientifically, this is one of those things that's fun to think about. Does anyone know why they use arsenic?
Because it was an ancient, Chinese treatment.
Right. So people with PML, right, and the myelocytic leukemia, it's not PML anymore, but people with that form of leukemia that has that genomic translocation also respond to arsenic, right? And there's something going on that people still don't understand that arsenic does to tether these PML bodies or perhaps strengthen their integrity. And really, it's known that the leukemia, the formation of leukemia, is associated with the disruption of these bodies. So there was this ancient Chinese concoction where if you had this disease, they would give you this concoction. And, who knows everything that was in it, right? It's impossible to imagine. But, it's one of those, take the concoction and start to purify it, start to separate its components and figure out what's the active ingredient in there, because something is really working. And the answer ended up being a poison, right? Arsenic. Which is good thing because we never would have thought of that. To give poison to cure leukemia. Although I guess most forms of chemotherapy are essentially poison. But I'm getting off track now. The point is that arsenic will stabilize these bodies. And this is really important too. I think maybe we'll skip to the data because we're about out of time. You got to have a graph, right? Because what they see is that when you stabilize these original bodies in a cell, number one, then either you can do it by arsenic or by proteasome inhibition, right? What you see is an attenuation of viral replication in those systems. So there's something about these pre-existing bodies that is important, right? And even here, they don't really look at the way they're thinking about this, right? And this is one of those things. So on, I think on Tuesday, the Indie speaker for Wednesday is giving this seminar at 4 o'clock, which is really right up the alley of this punchline, right? Which I think is how does the expectations of authorities, influence experimental design? And, to translate that to you, what it really means is how does what your PI think affect you when you swallow it whole and believe that must be true, right? Because when you get these lab groupthink hypotheses, many times they're right, but it's really worth thinking about how you could be tricking yourself by buying into the idea too fully, right? And, and not to throw these guys under the bus, I think they did really great science, but if you guys are interested in thinking more about it, the guy, he's published things in Nature about how to think about these things, and being sure you're thinking for yourselves, which I think is pretty important for graduate students, so maybe you can show up. But the bottom line here is they're thinking of like, oh, they have no PML there and it's not working, right? Kind of look at the way they're thinking about this. But really all the data says that if you stabilize these things right here, you prevent the subsequent aspects of replication, right? Now they're imagining that they're disrupting these bodies so that the protein can be dispersed so that they can come back to these new bodies that are formed that are really great for the virus, right? But maybe there's some antiviral potential here that the virus is well aware of that it's disrupting, right? Because when the last experiment just showed that when you stabilize those things, the virus doesn't replicate, right? But this was kind of before restriction factors were trendy, and the thinking was, okay, there's a virus, and its association with the host protein will include hijacking the host proteins to do whatever the virus wants to do. And that's not like that never happens, right? Before this course is over, you're going to see dozens of examples of when that's true, right? But the thinking of there's these specific antiviral proteins that sit in cells and wait for hell to break loose hadn't really caught on yet, right? There's only a couple examples of this in 2001. And so they're like, oh yeah, well these things have to be broken up to help PML get over here. Well, maybe TRIM19 is mediating this antiviral effect in these cells. Because there's, we could have done a very similar paper involving adenovirus and how adenovirus has to get rid of these things, right? Now, why is that? Now, no one really knows. And so TRIM19 is one of those things that to this day, people still don't know what it does, right? There's like a huge paper there somewhere, but kind of the exact mechanism that it's eliciting its effect is very unclear. But I think what's pretty clear now is that PML is mediating some sort of antiviral effect, right? And it's getting recruited to these big replication centers. And if anything, I tried to squeeze in a little host antiviral defense into this lecture. But the bottom line you should walk away with is that when you look at a cartoon, right, like we did in the beginning, and here's the mistake I don't want you to make. This just does not happen floating around in space, right? This just does not happen, if you look at it up here, right, these electron micrographs prove a point, but they can be so very misleading, right? This just looks like this thing is like a self-contained entity that is floating around in the space on your slide, right? But in reality, most viruses that replicate their genomes in a big scale have these compartments where inside the cell there's a very constrained reality where all of the factors that are required for this are recruited. And these are huge macromolecular assemblies. They're not just single molecules that you see in the cartoon, right? And that's at least worth really considering, especially when you're thinking about DNA viruses, that because of their huge genomes and the amount of genome, like when a 5 kb virus replicates its genome and it makes 10 to the 4th copies, you're like, all right, yeah, pretty good, right? But when something that's 200 kb makes 10 to the 4th copies of its genome, you have to tip your hat, right? That's a heavy lift, right? And it does that by utilizing structures like that. I guess that's all I have to say about that. Any questions? All right. I will see you guys all on Monday.
Are you sending us papers? Or we continue on?
Yes. No, we will. I think we're done with that. I will send out the paper on Monday. Again, so I think what I'm going to do is there's this really classic retrovirus paper that I want to go over, but try to read it. I think it's a good. And it's just one of those papers that's been referenced like over 100 times. Like if you studied MLB, like you couldn't not know this paper. But some of the methods are kind of old, and sometimes figuring out learning exactly how they did science in the late 80s is not a lot of value to you, because then you're like, well, when am I ever going to do that? Right? So I'll walk you through it again after we do some retroviral replication, but you don't have to be able to walk me through every figure.
So I want to know what kind of questions do we expect on exam on these papers and based on lecture?
Yeah, I find I'm very creative. So, I mean, what I would definitely say is I would expect less, fact-based things, at least for here. I mean, I'll probably tell you things about reverse transcription you should know, on Monday that I think really helps everyone understand things. And maybe there'll be a little bit of memorization necessary. But I think the big ticket items, and what I'll tend to do is I'll invent scenarios or viruses even, where I'll be like, oh, this is happening. How would you look at that, or how would you think about that? And so it won't be as much of a regurgitation of information, at least especially for the DNA viruses lecture, because there's not like retroviruses. There's retroviruses and HIV that we'll talk about later. So there we can kind of get into more detail where maybe some detail would be appropriate. But here there's so much going on, and DNA viruses do things so much differently that I don't think I'm going to pound you on specific proteins. Maybe some of the big ones, maybe the big ticket items that we talked about, but spend less time memorizing and more time thinking, and I think you'll be in better shape.